My name is Hana Jungová, and I'm a researcher at Chalmers University of Technology in the Department of Physics. My research combines physics, nanotechnology, and biophysics. And one of the research areas I'm focusing on is developing new innovative techniques to detect cellular motion on nanoscale. So think of the way how health monitors track our vital signs, for example, our heart rate or oxygen level. So in a similar way, we have developed a health monitor for single cells where we track the motion as a signature of life and health. So we are using gold nanoparticles, which are rotating very fast in optical tweezers, and by monitoring the change in the motion of the particle, we can determine the motion patterns of single cells. So for example, we can use this technique to determine the response of the cells to changes in its environment or response to different drugs. So we can, for bacterial cells, for example, determine how antibiotics are working in real time and if the treatment of this specific species of bacteria is effective. So by developing this tool, we obtain a new means how to monitor cell health and life, and we can apply it to research in various diseases. Our research opens up several possibilities how to develop new innovative tools for studying disease and in therapy and diagnostics. For example, in the case of bacteria, we can detect if the bacteria responses to antibiotics within just a few minutes. And this procedure, the antibiotic susceptibility tests, nowadays take several hours to days. So this could really speed up antibiotic susceptibility testing. And this is just one application area. Another possibility is to study how cancer cells react to their environment and how can we effectively treat cancers. So in the future, this can lead to personalized medicine and more effective treatments of various diseases. The grant from Hasselblad Foundation is instrumental for my research because it will allow me to establish an independent group in nanoscale biophysics, which has always been my dream. And it will allow me to create a place for young researchers who could join our team and expand our research area in the nanomotion research.
My name is Björkman. I'm a senior lecturer at the Department of Biology and Environmental Sciences at Gothenburg University. My research focuses on climate change effect on Arctic ecosystems, and especially on the carbon balance in Arctic plant communities. We have a rather good insight in what the vegetation will do in the future. Arctic vegetation is estimated to grow taller, change a bit in species composition, and also increase in biomass. But we'll know less about how this will influence the actual carbon budget. So basically how much the plants takes up in terms of carbon, and also how much the ecosystem and the soil below releases in carbon dioxide, for example, to the atmosphere. With a grant from the Hasselblad Foundation, we will be able to synthesize carbon uptake data from more than 50 experimental warming treatments around the Arctic. Experimental warming treatments basically mimicking a future climate, roughly 30 to 50 years ahead of time. And in this way, we can see into what kind of carbon balance we estimate the future will have. This research will help us understand the risk climate change poses on Arctic carbon balance, especially the uptake and the emission processes within Arctic ecosystem. And by this, it will help us to understand which mitigation pathways we need to take in the future.